thank you very much for the introduction and for the invitation to speak to you all tonight. Thank you to everybody that's joined and is joining. It's really fantastic to have you all here this evening. So my name's Christopher Stewart. I'm a research fellow at Newcastle University, which means that I basically run a lab and we exclusively do research. I'm a white male. I have short brown stubble, short brown hair. I'm wearing a green shirt and I'm actually sitting in my loft out the way now. So behind me is just the attic and white walls and a white ceiling. So as I said, really fantastic to have you all tuning in this evening, and I'll be talking about the microbiome, that is the bacteria that colonize babies, and the impact and the role that this plays in short and long-term health. If you've got any questions, just pop them in the chat as we go. I'm hoping to leave plenty of time at the end for discussion. So what is the microbiome? It's important that we understand that because it's probably the word I'll say more than any other over the next 40 minutes. Quite literally, it translates to the small community that occupies a distinct environment. And we all hear about one particular microbe on the news, and we have done over the last couple of years now in coronavirus. And there's a range of other human viruses alongside bacteria and fungi. And you may well know about fungi because if you, for instance, let your loaf of bread go a day or two out of date, you'll start to see the green mold growing on that. You might also be familiar as it's used in breads and in the production of things like beer as well for fermentation. But I will very much be focusing on the bacteria element of the microbiome, and I'll be focusing mostly on the gut or the intestinal microbiome specifically. So as we sit here this evening listening to the webinar, most of the cells that occupy our human body actually come from our microbes and not from our own human cells. And so in that regard, we're more microbial than we are human. Some 56% of the cells come from those microbes. When we consider that at the genetic level, over 99% of the genes encoded within the human body come from our microbiome and less than 1% from our own human genome. So clearly a vastly important contribution both at the cellular and the genetic level is being imparted by the bacteria and other microbes that we have colonizing the body both externally and internally. And I like to think about this in terms of macroecology. It's a lot easier to think about things that you can see rather than microorganisms that you can't. So if we think about the planet, and here's a map of the world, it's really not surprising why certain animals or plants or fish are adapted to the environment that they're in. So you think of a polar bear in the Arctic is clearly going to be a very different environment to the sort of mountainous regions of Canada or the vast desert plains of Africa and so on. And so you wouldn't expect to find a polar bear, for instance, in the vast plains of Africa. And equally, you wouldn't expect to find other animal species in other environments. And that's because they've developed very specific adaptations that allow them to thrive in these environments because they're suited to the terrain, they're suited to the food sources that are available, and they're also very well suited to things like the temperature and the humidity, etc. So if we think about that, also applies in a microecology theory as well. So the human body to a bacteria is very much like the world to an animal species. And so a very short space over the human body could literally be like flying from one continent to another. So if we take the example of the mouth, the microbes that you will find there typically will thrive in an environment that's quite moist, that is quite warm, and that would be then very different to what we would expect to thrive in the stomach or the intestine, where the oxygen levels are very low. And of course, there is a much higher nutrient source coming into that environment in the foods that we eat. And equally, perhaps not surprising to think, since the bacteria are using the food that we provide in the diet for their own growth and development, the microbiome of a vegan, for instance, would be very different to the microbiome of someone that eats meat. And they're quite large geographical spaces, if you think about it in terms of the world, using the human body as the analogy, but even on a very short proximal space. So let's talk about the fingertips to the palm of the hand. The palm of the hand would be very much equivalent to like the Grand Canyon with its very rigid and dry terrain, which would be different to the fingertips, which is much more of an oily marsh type of environment. And therefore, the bacteria that would exist on the palm of the hand compared to the fingertips would also be very different, despite being very close together. And actually, if I took each of your mobile phones off you and swapped and sequenced what bacteria were there, and also sequenced the bacteria from your fingerprints, I'd be able to pair up your own phone with yourself based on what microbes you've left behind on the phone screen as you've touched it. And this has even gone as far as to have forensic applications. So it was actually featured on CSI Miami, where they used the criminal's microbiome in order to place them at the crime scene. And of course, that is very Hollywood, but it's not unreasonable to think that that can move beyond Hollywood and actually into forensic investigations in the very near future. And whilst that's very exciting, I am more interested in understanding the role that the microbiome plays in infant health and also disease. But it is important to think, despite most of the headlines covering the negative aspects of our microbiome, for instance, COVID, the vast majority of microbes that colonize the human body are actually providing beneficial properties for us as humans and towards our health. So this is a non-exhaustive list of some of those features which are important in infancy, but quite frankly, throughout the life course. But one thing in particular is this education of the immune system. So after you're born and you're colonized by bacteria for the first time, those bacterial cells start to interact with your own human cells and your immune cells. And we call that the host microbe interaction. And over the first year of life in particular is when the immune system is really strongly developing. And that interaction over that first year of life can really seed very important features that will predispose infant to short and long-term health or disease statuses. And we'll unpick that a little bit further throughout the presentation today. I'm particularly interested in preterm infants, and I'll talk about those towards the latter stages of the talk today. But in particular, they will face challenges having been born early, having had a sort of weakened and immature immune system, but also being housed in a hospital environment in an intensive care unit for the first weeks or months of life. This will then basically seed them with a different microbiome to what we would expect in a normal term population. And this abnormal bacterial colonization, coupled to the higher amounts of antibiotics that are being used in that population, can predispose them to a range of diseases and disorders, including and spanning neurological disorders, cardiovascular, respiratory. But ultimately, where I focus most of my research is on infectious disease and intestinal diseases, in particular necrotising enterocolitis, which, as I say, we will come on to in the latter stages of the talk today. So the microbiome over the last decade or couple of decades really has received a remarkable amount of interest from the research community. And you can see that here plotted as either microbiome or microbiota. And that isn't because all of a sudden we discovered that bacteria are important and we need to understand more about them, but rather the technology that allowed us to understand what bacteria were present in a given sample or ecosystem have rapidly improved over the last 10 or so years. And as scientists, we're often led by the technology, and this is just a classic example of that happening. Technological development, coupled to reduce cost of using those technologies, has enabled researchers around the world to start investigating the role of the microbiome across a huge range of health and disease statuses. In fact, almost every disease at this point in humans has been studied somewhat in terms of how the microbiome might be associated with its onset and potentially used as a prevention or a therapy. So again, going back to understanding what we can see and how we learn from that and apply that to microecology theory. In the 1800s, Charles Darwin, who I'm sure many of you will have heard of, one of the pioneering scientists around evolution and natural selection, went to the Galapagos Islands and began to characterize, in particular, the bird species that he found whilst he was there. And what he noticed was, as he went from one island to another, the food source available to these finches or birds was very different. Some of the islands were particularly rich in buds and fruit, and others more insect or grub based. And he then started to notice that the beak shape of the finch very much reflected the diet that was available or the nutrient source available on that island. So birds that required the buds or fruit for their nutrients tended to have a much stronger beak for breaking down those hardier food sources, whereas those that were eating grubs and insects had much more sort of longer, thinner, more finesse types of beak because they didn't require the same level of strength. And this actually here on the right is a photo from his diary where he postulated for the first time the tree of life, and that is that we all derive from one common ancestor, and over the course of evolution, an advantageous feature that allows a particular species to thrive will be maintained, and less advantageous features will, over the course of evolution, be lost. And if you like, the plot here basically has A, B, C, and D, and he was using this to postulate that the common ancestor between A and D would be this point here. And you might think about the finches as sort of all being around this area, which would be very different to those, to potentially, for instance, humans or gorillas or apes, which would be situated over here. Technology, biology, science has improved clearly a lot over the last couple of centuries, and now we have a very well-documented tree of life. And a lot of it makes sense. You can look at a fish and understand why it's particularly well suited for the oceans and why humans are particularly well suited for land. But it becomes a lot more challenging to understand the biology and why some particular bacteria and other microscopic organisms thrive in environments when you can't really see them. How do you begin to then characterize them? Traditionally, this was done using culture and microscopy. So here's an example of just how small these microbes are. This is the microbes on the end of a drone pin. Of course, you'll never see that by eye. One way we do it is by growing on what we call agar plates. These are nutrient-rich jelly-like substances. And actually what you're looking at here on the right of the slide is my handprint. And I assure you, I don't have especially dirty hands. Every one of us will have a unique microbial fingerprint, as I said earlier, and this is mine. And so you can see a range of different bacteria here as these different colored yellow and white dots. And you can also see some fungus, probably penicillin, growing here also from my fingertips. And this is a fantastic tool for having these bacteria in the lab. And of course, we can test these now to see if they will grow in certain nutrients or not. And there's a lot of experiments you can do, but it's very hard to identify what they are. And so that's where sequencing has really came in. And this is why over the last 20 years, the number of articles involving microbiome has gone up exponentially and continues to do so. I won't go through these in detail because it's not really important to the overall understanding today, the ins and outs of the different methods, but principally just to mention there's two techniques. One is the 16S or RNA gene sequencing approach, and the other is metagenomics. Both techniques sequence the DNA that we extract from clinical samples. For the purposes of my research, that tends to be poo samples or stool samples as a proxy for what's going on upstream in the gut. And the biggest difference is that this approach uses an amplicon step, so a particular region of bacterial DNA is amplified and then sequenced, whereas in the metagenomic approach, all of the DNA in a sample is sequenced. So we'll have human DNA, bacterial DNA, fungal DNA, virus DNA, etc. The added benefit of metagenomics is also that we can get information on what genes are present. So not just identifying the names of bacteria, but ultimately what genes and potential function those bacteria have. And so we can do this at an unprecedented scale. We can extract hundreds of samples in a day. We can sequence those samples overnight, and the next day literally have information from hundreds, if not thousands of samples. And then those bits of DNA are mapped to databases, and because these databases are very well annotated, we know that this particular sequence is from an E. coli, this particular sequence is from a different bacteria, etc. And this is what a typical microbiome or omics study might look like. So as I said, our input to most of our studies are feces or stool or poo samples, but of course there's no limit there. You can also look at urine or blood. They aren't necessarily the best samples for microbiome studies because they're typically sterile samples, but could be useful for some of these other omic or technologies. So genomics, of course, is the sequencing of DNA, and that could be also taken one step further to look at transcriptomics, which is the sequencing or sequencing the gene expression, all the way down to metabolomics, which is the final small molecule that is being produced by these cellular reactions. And of course, the more technology you can apply to a single sample, the more layers of information you can understand, the more pieces of the jigsaw puzzle you have. But this is expensive to do a lot of this work. And so usually you might pick one or two that you apply to the samples. The sample or the data then undergoes a range of quality control and data analysis, which ultimately leads us towards biomarker discovery, allowing us to have predictive models, and perhaps even the development of novel therapeutics. And I'll talk a little bit about advances we've made in that area in a few moments' time. So let's get into the data. And one thing that's really captured my interest is this finding and the whole hypothesis around the hygiene hypothesis. So this is now 20 years outdated, but the trends very much still persist today. And that is that the incidence in the Western world, at least, the incidence of infectious diseases is declining rapidly, and this is amazing. However, this is coupled to an really unprecedented rise in the incidence of immune disorders. So for instance, Crohn's disease, type 1 diabetes, and asthma. And so putting these two things together, the hygiene hypothesis suggests is we've got cleaner, we've got more sanitary, we're using more disinfectants in the environment, we're using more antibiotics than we've ever used. We've been able to get on top of a lot of these infectious diseases. But the consequence of that is we've altered our microbiome vastly, and this has caused negative consequences in how our microbiome and immune system interact with each other, and then ultimately is causing this rise in the incidence of immune disorders. At the moment, it's only a hypothesis. There's some evidence to suggest that it may well be linked to the microbiome, but certainly his work is very much still ongoing. But one study we sought to use to address this was called the TEDDY study. So TEDDY stands for the Environmental Determinants of Diabetes in the Young. So we were specifically looking at the diagnosis of type 1 diabetes. This study sequenced over 12,500 stool samples using both of the sequencing technologies I mentioned earlier. And these were collected from people who were living in America and also Europe. And in total, over 900 infants were included in the study. So most infants give multiple samples, and we followed them really from birth until around month 46 of life. So this is published. It's actually open access. If you're so inclined after today's talk, you can go away and read the study in depth. But it featured on the cover of this journal here, this scientific journal called Nature, which is really cool. And you can see the imagery here is trying to depict that we have a teddy bear to represent the TEDDY study. Of course, it's involving babies and infants, and wrapping around the baby's intestine here is what they call the security blanket. And you can see these pathogens lurking here in the bacteria, providing protection to the infant. And taking center stage in that depiction is these Y-shaped bacteria, and they're called bifidobacterium. And I'm going to come back to bifidobacterium a couple of times throughout the talk today. So with such a ridiculously impressive data set, one of the first questions I was keen to ask was, taking all the factors that we think are influencing the microbiome development over that first year of life and beyond, what are really the main drivers? So we collected a range of information about the mother and about the baby and about its environment, all the way to what it was eating on every day, whether it lived on a farm, had fluffy pets, lived with siblings, etc. The most important, or what we felt to be the most important, were taken forward into this analysis. And what you'll see at this first time point, when we basically ask the algorithm what factor is driving microbiome progression or development, it was very clear that infants who were receiving breast milk had a very different microbiome to those that were not receiving breast milk. And this was driving microbiome development at this time point. That was true all the way through until 1 year of life. I'll stop the animation here, and you can see again, breast milk is really driving microbiome development at the 1 year time point, as well as things like birth mode. So whether you're born by vaginal or cesarean delivery, and whether you were in Europe or America, or whether you had siblings in your household, all had some impact, but breast milk by and large was the most predominant driver of microbiome. After 1 year of life, as the WHO guidelines state, breastfeeding is usually not taken. Infants no longer receive breast milk, and we no longer see that as having a significant impact. And quite frankly, by this point, the microbiome of an individual has become very individualized and is really no longer influenced by any of these historic or current events. So that's looking at which bacteria are present, but let's now turn that to what the function of those bacteria are. And when we do that, we see actually only breast milk is having an impact on the bacterial function. And again, that was only over the first year of life and no longer after. So from this, it's quite reasonable to conclude that receiving breast milk is driving an infant's microbiome, is very different to an infant that doesn't receive breast milk. And some of these other factors are important, but to a much lesser extent. And this, for me, was very striking because we know from a huge range of studies now that breast is best when it comes to the nutrition for an infant. So there's 2 examples here, and there's hundreds more in the literature which show epidemiological studies where an infant has received breast milk in their first year of life or not. In those infants who have received breast milk, they typically have reduced risk of developing diseases like allergies, asthmas, obesity, etc. And that's not just in the short term while they are being breastfed. The positive impacts of breastfeeding on the infant are sustained throughout the life course. So this is obesity in children, and we see impacts all the way into adulthood. So something is happening whilst an infant in the first year of life is receiving milk that's really programming them towards a much more healthy state throughout the rest of their life. And as I've just mentioned, breast milk is also heavily impacting the microbiome. So one wonders if this lifelong protection really boils down to the change in the microbiome as a result of breastfeeding and the subsequent then different interaction that it has with the host and the host immune system. I should just say here, because this audience, I want to be clear that this is all relative risk scores. So this isn't to say that if you are breastfed that you're guaranteed never to develop allergies or asthmas, and equally, just because you may receive formula only doesn't mean that you're guaranteed to go on to develop these diseases by any stretch of the imagination, but this is an overall relative risk. So the next obvious question, I've said about it changing the microbiome, but what about the microbiome is actually being influenced? And this brings me back to the bifidobacterium, this specific bacteria, which we've known for quite some time to be a beneficial microbe. Above everything else, when we did our modeling, it was very clear that as an infant is breastfed, they have a really high abundance of bifidobacterium. And as breast milk is removed from the diet and is replaced with formula and solid food, those bifidobacterium start to decline. So why is that the case? What is it about breast milk that allows these bifidobacterium to thrive in the infant gut? And we've done a lot of work, and as have others in this area, and this is something that I'm incredibly excited about. So there are sugars in breast milk called human milk oligosaccharides. I will abbreviate them to HMOs for the rest of the talk. And these HMOs are the 3rd most abundant component of mother's breast milk. They're more abundant than things like protein. Of course, protein is absolutely essential for the infant's growth and development, yet these sugars are more abundant, suggesting potentially more important. That being said, they can't be used by the infant for any energy or nutrific value. Babies lack the enzymes to break down these complex sugars. That means they reach the small and the large intestine fully intact, and it's there that they provide a food substrate for the bacteria, in particular the bifidobacterium, which have the genetic repertoire to use these sugars for their own growth. So that's not to say they don't have any other impacts as they're passing through the intestine of the infant, but certainly it's not nutritional. And for that reason, we consider them what we call prebiotics. That is, that they're providing a substrate to promote the growth of beneficial bacteria. And here is a bifidobacterium down a microscope. As I say, it's this really cool Y-shaped bacterium, really fascinating to study. And if we take a look inside a bifidobacterium, we can really start to understand why it might have potentially beneficial roles in an infant gut. So here we have the human milk oligosaccharides. We know bifidobacteria can take them intracellularly. We know they have the enzyme complexes to break down those HMOs. But what's really amazing is that the byproduct of this reaction, if you like, the waste product that the bacteria doesn't want, is what we call short-chain fatty acids, in particular acetate and lactate. Short-chain fatty acids we've known now for a long time to be important to human health and human development. These short-chain fatty acids can be uptaken by human cells. They also interact directly with the immune system. They have anti-inflammatory properties and a whole range of other features that we know to be beneficial. And particularly when we think of inflammation, that's going to become even more important when we think about a preterm infant for reasons I'll go on to explain. So here's one potential, what we call mechanism, whereby the provision of human milk oligosaccharides through mother's milk actually increases the growth of a bacteria that basically spits out a waste product that's incredibly useful for that infant to have. So it's important to understand why these HMOs have existed at such a high abundance in the milk. As I mentioned earlier, evolution doesn't mess around. If something was non-advantageous to an infant, such as these complex sugars, the mothers would simply stop making them in the breast milk at the levels that they currently do. So the fact that they have been through the course of evolution and continue to be so abundant really, really does highlight that this interaction between the nutrients in the milk, the growth of the bacteria, and the interaction with the immune system are really critical over that first year of life. This has recently been taken one step further. So you mentioned now the human milk oligosaccharides present in the mother's breast milk aren't metabolized by the infant, so they reach the intestine intact. They promote the growth of these different bifidobacterium. So you all know that now, and you know about the short chain fatty acids, but there's another compound which is also very important to an infant, and it's just in the last couple of months been shown that these bifidobacterium can also convert aromatic amino acids into aromatic lactic acids, and these play very important, what we call immunoregulatory roles. So not only do the bifidobacterium give us short-chain fatty acids, but they also produce indole lactic acid as well as some other lactic acids. And ILA, indole lactic acid in particular, again, has anti-inflammatory properties, which again is vastly important. We unless it's absolutely necessary, we really don't want the intestine to be inflamed. And in a preterm infant, as I'll go on to talk about, we really don't want inflammation in the intestine for as much as we can avoid it. So here's a few mechanisms whereby diet is changing microbiome and is impacting health directly. So in the last 10 minutes or so, I'll really dive into some of our preterm work. This is really the focus of my lab now at Newcastle. I'm fantastically privileged to work alongside Janet Barrington and Professor Nick Embleton, who are both neonatal consultants in the RVI, the Royal Victoria here in Infirmary here in Newcastle. And indeed, my research laboratory is literally a 5-minute walk from the neonatal intensive care unit from the NICU. So I can get to the patients, to the babies, to the samples without ever leaving the building, which is unique. I don't know anywhere in the world that's as well set up for this type of work as we are. And so when I talk about a preterm infant, I'm specifically referring to those that are born between 22 and 32 weeks at gestation. To put that into context, a 22-week gestation infant is really just over halfway through a pregnancy. So you can imagine they're expecting to be in the womb for the next 40% of their development, but all of a sudden they find themselves born. And medical advances, massively improving over the last 20 years, have allowed us to keep even the most fragile preterm infants alive. But this comes with increased challenges because the immune system of the infant hasn't fully developed, the intestinal architecture of the infant hasn't fully developed, etc., etc. But what it affords us the opportunity to do is to collect a range of samples. And this is a list of some of the samples that we routinely collect from this population. Of course, these are all non-invasive samples, and we, in the instance of, for instance, breast milk, will salvage only what's left in the syringe once the baby's been fed. We will obviously never take a breast milk sample that a preterm infant needs purely for research. So it's very much a salvaging operation. Similarly, with the more invasive samples around blood and tissue, we will only ever collect what's clinically indicated and needed to happen. And indeed, we'll only actually collect what would otherwise go in the bin. So Really, we are very well set up to salvage and maximize how much we can learn about these diseases by the mechanisms that Janet has worked very hard to put into place. And all of these samples, we now have over 100,000 samples from this population, and they're all housed in our Great North Neonatal Biobank, which is open literally to the world and to all researchers to come and use and really hopefully advance our understanding of preterm disease. So I mentioned earlier necrotising enterocolitis. This is something that's captivated me now since I did my PhD 12 years ago. It's a disease that continues to be the leading cause of death in this population as more and more preterm infants are surviving being born prematurely. The risk and the numbers who are succumbing to this disease increase all the time. And if nothing else, in my life's work and my research, I want to make huge strides in our understanding and hopefully our ability to prevent or reduce the disease in preterm infants. The disease itself is just an exaggerated inflammatory response. As I said, these are incredibly immature infants. The more premature the infant is, the more at risk they are of developing the disease, simply because their immune system is immature. The cells lining the small intestine and the large intestine haven't quite formed their tight junctions, so they don't fully bond to one another, and we call this leaky gut. And all of these factors combined basically mean that the preterm infant starts to respond in a very exaggerated inflammatory way. Long story short, that reduces oxygen to the environment, which to the detriment of that tissue then becomes necrotic. And we can sometimes medically manage this with antibiotics and controlling feeds, but more often than not, the infant will require surgery to have that region of their bowel removed. And of course, this will have significant lifelong morbidity for that individual. But over the last 10 years, there has been promise from work involving human milk oligosaccharides and the microbiome. And I think we have been at the forefront of that in Newcastle, but there's many others around the world who are also doing fantastic work. And frankly, we've never understood more about this disease than we do now. So here's one study. This was led by my PhD student, Andrea Massi. It was in collaboration with Professor Lars Bode, who's based in San Diego. And we collected milk from mothers prospectively and then retrospectively matched 33 neck infants to 37 controls. And the first thing we asked was, are these sugars, these HMOs in the mother's milk, different between those infants who go on to develop disease versus their healthy control counterparts? And what this plot is showing here, each circle is a sample, and it's very clear that those infants who went on to develop neck are receiving milk that has different sugars compared to those infants who were healthy. We then asked what sugars are different, and strikingly, it was one single sugar called di-siallacto-N-tetrose. I'll abbreviate that to DSLNT for the remainder of the talk. And this particular HMO was higher in our control babies compared to those infants who went on to develop the disease. So this has obviously given us a potential predictive tool. It could be used as a biomarker, but it could also be used as a therapy, indeed, if these kind of findings hold up when we do further work. So we're incredibly excited about this. We use some modeling, some quite simple modeling, frankly, to determine what the optimal threshold for this particular HMO would be. And we said that 241 nanomoles per mil, that's this blue line here, using that as a threshold, we would be able to identify 90% or 9 out of 10 infants correctly as either being control or at risk of necrotizing enterocolitis. And that would be crucial information to a clinician, because this disease can literally manifest over an incredibly short space of time, by which point it can be quite hard to manage medically. So if we have a ability to predict those infants who are potentially at risk, that would really be a game changer in the clinic. What's also really nice, and frankly very rarely happens in science, is this corroborated previous work from the collaborator Lars Bode, who showed that control infants had higher DSLNT compared to Bell 1 is a kind of low severity neck, and that was significant. But more importantly, those individuals that went on to be Bell 2 and 3, this is our more severe form of necrotizing enterocolitis, you can see that the DSLNT levels in their mother's milk again was significantly lower than the matched controls. So 2 separate studies at separate times with separate cohorts deriving at exactly the same results. It very rarely happens in science, but is especially exciting when it does. Lars has actually done some really nice work in this area. This is using a rat model. Here they either fed the rats the dam fed, so kind of mother's milk. They also used formula milk, but added human milk oligosaccharides. They also added formula milk without human milk oligosaccharides. And you can see that those infants who were receiving mother's milk or formula with human milk oligosaccharides had much higher survival, somewhere in the region of 195% survival, compared with those infants only receiving formula, not supplemented with human milk oligosaccharides. Their survival was significantly lower. And something that I really like that the team did to take this one step further, because it appears that this DSLNT sugar, which is structured like this for any biochemists out there, appears to be very important and very specific. So the team then asked, what if we start to manipulate the structure very slightly? Will that impact the ability of this HMO to reduce disease? And indeed it did. So you can see here DSLNT, the pathology score is less than 1. 0 is normal, 4 is complete destruction. Anywhere below 1 is very good. You can see here DSLNT can reduce pathology compared to formula or formula supplemented with DSLNT-like structures. In those cases, you see much higher pathology scores. So again, some mechanistic proof that there's something intrinsically beneficial about that particular structure. We're a microbiome lab. I feel like I haven't mentioned microbiome for a while, so I have to get back into my comfort zone. And here we followed up that cohort of infants where we had the maternal milk, and also looked at the infant stool samples to understand what microbes they had in their intestines. Here we use an algorithm that basically says, here's 5 different clusters, and if a sample had high Staphylococcus, a particular bacteria, it would be in cluster 1. If it had a lot of Escherichia in its sample, it would be put into cluster 2. If it had a lot of Klebsiella, it would be put into cluster 3. Cluster 4 would be samples that were dominant in Bifidobacterium longum and Bifidobacterium bifidum. And finally, cluster 5 would be dominant in Bifidobacterium breve. And so this is just a way for me to take a cohort of thousands of samples, very complex, and actually just distill it down into 5, what we call preterm gut community types, or PGCTs. And I number these clusters 1 to 5 based on the average age within them. So this doesn't tell us anything, but it's a means for me to be able to do some more statistical analysis. And so that's exactly what we did here. These plots look a little bit confusing on the surface. I'll just spend a moment to sort of go through it. So here's the 5 preterm gut community types I just mentioned. Really, the only thing you need to remember is that preterm gut community types 1, 2, and 3 had low bifidobacterium, and clusters 4 and 5 had high bifidobacterium. Remember, bifidobacterium is our beneficial bacteria. Generally, the more of that we think, the better in this population. And then as we move through time here, these are the different day of lives, DOLs. You see that infants start in cluster 1. They then generally transition into cluster 2, but also quite rapidly move towards clusters 4 and 5, or bifidobacterium-rich microbiomes. That's what we want. This is the healthy development. And so that's probably what you'd expect from those infants who were generally healthy. If we contrast that with those infants who were below that DSLNT threshold, so their mother's milk didn't have enough of that particular HMO, they didn't show that same progression towards those bifidobacterium-rich clusters that we really want to see. And so this is an extra layer, if you like, of evidence to suggest that we have this 3-way interaction between diet, microbiome, and human health. And so this was published in December last year, and is work that we're now actively following up to really begin to think about biomarkers and therapeutic strategies to use this DSLNT finding. And some work that we did actually a couple of years ago now also supports everything I've just talked about. So here, each dot is a different poo sample. The closer together 2 dots are, the more similar those microbiomes are, and the further apart 2 dots are, the more dissimilar those microbiomes are, and they're simply colored according to what the more dominant bacteria was in that sample. And so I'll point you specifically towards the yellow here, the bifidobacterium. And in a second, I'll play an animation, and you can watch an infant develop microbiomically through time, both a control and an infant who went on to develop NEC. And so I'll set that away. And immediately you can see our infant who developed disease has a very chaotic development of their microbiome. They're moving vast spaces across this plot, suggesting from one time point to the next, the microbiome is changing drastically. They also spend very little time in that yellow region, or bifidobacterium-rich environment. If we contrast that with our control infant, they start at the same place microbiomically with the staph dominant group, and that's quite common. They then swiftly move into the bifidobacterium dominant group and remain in there for the remainder of their sampling, moving very short distances. And so as you can take away from this, and as the paper, and again, it's open access, if you're interested, go and read it, as we show in that manuscript, infants who develop NEC have significantly less stability and significantly less bifidobacterium. So I'm really excited about all of this work. I think we have a prebiotic in DSLNT, we have a probiotic in bifidobacterium, a beneficial bacteria that might reduce disease in this population. In the last minute of the talk, I just want to mention where we're taking this to try and understand more about the underlying mechanism. And so I mentioned earlier about how when an infant goes for surgery, we will collect whatever would otherwise go in the bin. We can then isolate the stem cells from the base of the crypts of the intestine. These are stem cells that are proliferative, so they will continue to grow and divide. And if we provide the right factors, they actually become mini guts again. So we will, in effect, grow the patient's intestine in the lab. And this is an incredibly powerful model because for the first time, we don't need to use any animal models, and we can actually add bacteria and other stimuli directly to patients that we know have the genetic, the epigenetic risk factors associated with these diseases. To take that one step further, whilst I was in America, much more of an engineering and a biology project, we actually developed a system that allows us to control the oxygen that is exposed to the human cells, which you can see in this line here, and also the bacterial cells, which we sit on top of the human cells. And that's important because, as I mentioned earlier, the gut is an environment that has no oxygen. Bifidobacterium would die if there was any oxygen. So we can actually do this by keeping an anaerobic environment for the bacteria and allowing the oxygen to feed the human cells so that they survive and remain viable. So a really cool system. We're doing experiments with this model now all the time. I won't go actually through it because I'm keen to leave time for discussion, but just to say, as a very early bit of evidence, we do see a difference between preterm and adult derived organoids. So we do feel that we have here potentially a very robust model for exploring all of the things I've just talked about much further and really getting to the bottom of the mechanism of action then in these diseases. So I want to say a massive thank you to all of the people that's been involved in the work, in particular those from my group and the wider university here at Newcastle, and frankly, Janet and Nick, without whom none of this work would be possible, and then to our other collaborators, both in the city and around the world. None of this would equally be possible without the fantastic support of our funders and our national and local charity for NEC, and also for Tiny Lives based at the RVI. My email address is here, my Twitter's here. If you're interested in the subject, feel free to follow. I often tweet new positions that are coming up or interest in research in this area. And we also have a website, so we have blogs, publications, presentations, etc. So if you're interested, again, feel free to check that website out. So I'll leave it there. We've got plenty of time for questions, and just say again a massive thank you for the invitation and to you all for listening. Thank you.
Excellent. Thank you very much, Christopher. That was really, really interesting. And he pitched it perfectly for me, who doesn't have a science background. So that was amazing. It's really, really interesting to actually see some of the theory behind the breast is best kind of motto. You know, as a parent myself, you hear it from a midwife, but you don't really understand why. So no, that was really good. Thank you. We've had lots of questions, so we'll get through as many as we can. The first one, I'll fire away. Are babies' microbiomes affected if the mother had to have antibiotics, for example, if they've had mastitis?
Yes, very good question. And the short answer is yes, probably so. We know that one of the first seeding events of an infant occurs during birth and not before, although there is quite a lot of controversy in the field now about whether an infant is actually colonized in utero, but personally I think that colonization event in the vast majority of pregnancies happens at birth. If that mother has had antibiotics in the recent history, their vaginal microbiome, skin microbiome, but more importantly their intestinal microbiome is likely to be different. And one of the major routes of colonization through vaginal birth isn't actually through the vaginal tract, it's through the fecal matter that the baby also ingests during delivery. And so if a mother's had an antibiotic and it's changed their intestinal flora, the bacteria that colonize the infant after birth is also likely to be different.
Thank you. And a question about your route into research. So as a research fellow, how do you know where to start with your investigations? Big question.
Yeah, it's an expansive question. My route into research was very non-deliberate. I was planning on playing for Newcastle United football until I was about 17, and here I am. But frankly, love what I do, and I'm delighted that I've sort of ended up where I have. So as a fellow, the way it kind of works in a nutshell is I've now made the transition into independence. I no longer have a boss. I sort of am the boss of the group, and that comes with a lot of pressure because, of course, you have to have ideas and write grants and have people buy into your ideas. And so with my recent fellowships, it was really a case of sitting down with a blank Word document and really starting to read the literature and just giving myself the headspace to think about what would be an exciting project to do. Ultimately, I'm going to have to deliver on all this work I've now got funded. This is multiple years of work. If I wasn't invested in it and I wouldn't enjoy it out, it wouldn't be fun to do. So I'm trying to think what I would enjoy and what's really going to advance the field. And I build my background into that application, and then I go for it, because at the end of the day, I've got to show the funders that I'm capable of delivering the project as well as just thinking of a good project. So a lot of it's serendipitous as you go through. I'm trying to keep this answer short, so I'll wind it up now. As you go through like your career through an undergraduate, I never knew really that I wanted to study microbiome in infants. PhD opportunity came up in this area. It fully grabbed my attention, and then after that, I managed to go to a university in America that were developing organoids. Again, that was rather by chance and luck. But what I've then been able to do is build on all of the experience I've had to put into why I'm uniquely positioned to do the research that we're doing. So it just sort of writes itself in the end.
And thanks very much. And a question relating to COVID. So do you think infants born in COVID times would be affected by their limited exposure to everyday bacteria as they're kept at home and places are generally more sterile?
It's a very good question. We don't know yet, I think, is the short answer. It's too early to say, but I would speculate that is correct. So the person that asked the question sort of suggested that they would have a reduced diversity of microbes because they're not being exposed to daycares and schools and all the rest of it. And I would tend to agree. So I think that would have at least a short-term impact on the microbial diversity of their intestine and other body sites. But it's very quick to recover. So even a 6 month or a year out of these constant interactions with other children and other environments can still, we could, as soon as they go into daycare, they're going to eventually get exposed with all of the weird and wonderful bacteria that exist around these sites.
I can vouch for that with a toddler that went back to nursery, and lots of colds have started since. And the next question is asking about, does the microbiome have anything to do with why infants can't eat honey?
In some cases, possibly. So there's links with a whole range of things, celiac disease and a range of intolerances. It's hard, and I don't think there's any evidence to suggest honey specifically. So it's multifactorial ultimately. If the microbiome's involved, it may be just something's happened in early life, the immune system's developed differently, and now it's more of an immunological rather than a microbiological. But that's not to say the microbiome's not important, but it's incredibly hard to wean a specific intolerance like honey out from everything else that's going on, because there'll be a lot of confounders. So I'm sorry I can't answer definitively, but I guess it's possible. But at the same time, I don't think there's any work to definitively say that.
Excellent, thank you. And what developments do you expect or wish to see in the coming years in the study of the microbiome in terms of illnesses and treatments?
Yeah, so from a research point of view, I think we've done a lot of stamp collecting over the years. I'm not saying that in a derogatory way. We've learned so much about what bacteria now exist in these different ecosystems, but now we really need to understand what they're doing. So simply saying that these different bacteria are here without understanding the function of those microbes, I think, is very limited. So organoid work, etc., is hopefully going to give us much more clear answers towards that regard. And ultimately, the hope here, of course, is that we can start to manipulate and not just have a biomarker for disease, but actually start to give babies, in my case, probiotics. And we are now doing that. And I probably should have mentioned this. Because of the work that we've done and others around the world, in Newcastle, we routinely give all preterm infants bifidobacteria probiotics. So these are deliberately administered, viable, alive bacteria that we hope are going to improve health outcomes in that population. And that work has just started now. We need to understand exactly what species to be adding to these probiotics. But as you'll understand, the clinicians are desperate to improve outcomes in these populations. And we're very, very keen and supportive towards bifidobacterium and potentially other bacteria being able to do that.
Lovely, thank you. And is there a way of them being able to add these things, these microbiome, to formula milk?
Yeah, well, already that's happening. So less so about the bacteria. That's usually given as a separate product, but you can go to Boots tomorrow and buy a probiotic off the shelf and probably multiple different options, and you just sort of pick the shiniest packet or something, because there's no real evidence for why you would do that as an adult. And frankly, if anyone was to ask me about Yakult or Actimels or these sort of viable bacterial products, then as an adult, I would say you've got limited benefit from having it, unless you maybe is undergoing chemo or having antibiotics. Otherwise, it's just not going to colonize. Your microbiome is pretty well set in as an adult. But as a baby, of course, that's really a window of opportunity there to start to put in some of these good microbes. So probiotics are being used and are gaining a lot of attention, but also now formula companies are beginning to add these human milk oligosaccharides to their formula product. That might help, but breast milk is incredibly complex. I've picked a tiny little part of breast milk and talked about it today, and I haven't mentioned the other 99.999%. So you'll never get formula to be breast milk, but of course, any improvement is a good improvement. The problem you have is what we think are the most important sugars to add are also the most expensive to synthesize. So they're not the ones being added currently. Typically what's added is the more abundant, and they're not necessarily the best to have.
Excellent, thank you. And a question about why the geographical location affects the development of a microbiome.
Yeah, that can be a range of things, and frankly is probably confounded quite heavily by the different populations of people that are there. So for instance, in our TEDdy study, we had Germany and 2 sites within Scandinavia, and you'll expect the diet of Scandinavian infants and adults, which then seeds the infant, to be vastly different to even Germany. But then when you consider that with America, we had some southern states which had a much higher percentage of, for instance, Hispanic populations, whereas our Scandinavian sites were mostly white Caucasian. And so we classify that as geography, and it's a very good question, because the reality is it's everything else to do with that environment, and typically the ethnicity and the background of the individuals and the diet ultimately that those individuals are consuming.
Excellent, thank you. And I'll move on to a next one. That's exactly the question we've just said. Are there any ways that companies selling pre-formulated milk have made it possible to stimulate these? Oh, I think that's the same question we've already had. Sorry, ignore me. We've had a lot of the same question there in a row.
That's good.
This one. Why would the parent's BMI affect the baby's microbiome?
There's a range of reasons. So typically this might reflect the diet of the mother. So a higher BMI may well reflect a higher fat diet, of course, not only because the exercise is going to be an important contributor to BMI, but it's probably mostly a reflection of the diet that the mother has. Therefore, the potentially a little bit of genetic, but probably more diet, and ultimately then the microbes that it seeds, potentially the diet that the infant is then fed, will be different to those of lower BMI. It's a good question. That's more speculation. I suspect it's the case than any strong evidence, because we didn't really look at that further. But that's what I would think.
Excellent, thank you. And there's a question asking about what your undergraduate degree was before you went to your PhD.
Okay, so I did biotechnology. I picked that because on the deadline day of UCAS, it sounded really cool, and I was reasonably good at biology, and I quite liked IT. So I felt it combined the two. I would be lying if I said I had a great time as an undergraduate student, but what it set me up to do is been incredible. I've loved every second of being a PhD and then the researcher since. So I would say to put a bit more thought into it than I did, but yeah, also, like, I guess in undergraduate's hard. I found it very hard to learn a lot about a lot. Whereas like doing research is much more in my wheelhouse, because you learn about one subject that you're deeply passionate about at a very, very detailed extent. And there's a lot of problem solving as well. So as a researcher, I'm no longer in classrooms. I very rarely read books. Probably, frankly, don't read as many papers as I should, because I'm in the lab having fun and finding new things out. So yeah, the undergrad set me up very well for what I've now gone on to do, but it wasn't necessarily a very conscious and deliberate decision to get here.
Excellent, thank you. Quite an interesting question, this one. Can a baby receive breast milk from a different mother? What effect would this have on the baby's development? Excellent
question. So as we've learned over the course of I'm talking about preterm infants specifically here, but could be extrapolated to term infants as well. We know that one of the issues around formula milk is that it's from bovine origin, and the proteins from cows are different to the proteins from humans, and that causes the baby to respond negatively. And that's actually why formula feeding is one of the highest risk factors for disease in preterm infants. So now what we've just started doing in the unit earlier this year was using what we call donor breast milk. This is from mothers who have, either for free or for a small amount of money, donated their breast milk. And that's then pasteurized, so the bacteria that are in the milk are removed. That might have impacts on some of the other structures, but probably not the human milk oligosaccharide. And so the reason I love that question is because one thing that fascinates me is, can we start to now screen donor breast milk that's high in that DSLNT sugar and preferentially give that to preterm infants? Now, that's not being done, but I would love to think about that, and that would be one very easily translatable ways of improving the sort of clinical care of the infants.
Excellent, thank you. And two questions left before we end. First one is, does eczema affect the microbiomes on the hand?
We actually have a study looking at this in adults at the moment. It's hard to say directionality. So is the microbiome driving disease, or is the disease changing the microbiome? I am not an expert in that particular area, although, like I say, we're getting into doing a little bit of work in it. So I would hate to speculate one way or another, but the microbiome may well be different, but that doesn't necessarily mean it's causing disease. May just be a result of the disease.
And final question. Have you got any advice for any budding researchers out there?
I've got a lot, but we don't have time for all of that. Main advice would be find a subject that you're incredibly excited about, and realize that you will be learning something that nobody in the history of the world has ever known. And for a small moment in time, only you will ever know that. And then, of course, you'll tell everybody in the world will know. But I think that being on the cusp of that kind of understanding is especially thrilling, because on the day-to-day, and as I said, we work with poo samples. So literally we have freezers full of poo, we extract poo, we sequence poo, poo. So you forget sometimes like what the wider context of your work is, and sometimes it's important just to take that step back and realizing why you are surrounding yourself in all of this fecal matter is literally trying to save babies' lives, or adults, IBD, whatever it is. Find your passion, and the rest will just fall into place.
Fantastic. I think that's a really good place to end it. So thank you very much for giving up your time tonight and for such a fantastic talk. And thank you to everyone watching in the audience. We'll be back again next week, 6 till 7 p.m. on Thursday. So thank you. Enjoy your evening, Christopher.
Cheers.
Cheers. Thank you.
Thanks.
Bye.
Bye.